More than 100 million people worldwide are carriers of the trait sickle cell anemia. This disease directly affects 100,000 Americans. Here's a story on how it started.
A long time ago, in the villages scattered through Africa and the Middle East, disease spread like wildfire and medical treatments were unreliable. A man who had contracted malaria made a full recovery and seemed to be more resistant than the other people within the civilization. This man had developed a random mutation that made his cells sickle-shaped and his blood resistant to the infectious malaria. This mutation became an adaptation. He was able to pass this gene on to his children, giving them this advantage over malaria and a better chance at survival.
Even though millions of people are affected by this disease, not many people know much about it. Sickle cell disease is characterized by the sickle shape of the red blood cells. The sickle cell trait does not prevent infection from the parasite, but it does prevent the disease from taking hold after the host is infected. The gene stimulates the production of an enzyme called heme oxygenase 1 that breaks down heme in the blood, which releases small quantities of carbon monoxide. The carbon monoxide is the key factor for not allowing the parasite to take hold. Sickle cell disease is caused by a mutation in HBB, which results in a change in one of the bases in the DNA sequence from adenine to thymine. This then changes the amino acid in the hemoglobin protein from glutamic acid to valine and causes the body to produce a new form of hemoglobin called HBS. HBS causes red blood cells to develop abnormally and become sickle-shaped rather than donut-shaped. The sickle shape makes them harder and less flexible, which causes them to get stuck in the blood vessels, resulting in blood clots.
The gene we will be focusing on that causes this is HBB. HBB is short for hemoglobin beta. It provides information for making a protein called beta globin, which is a subunit of the larger protein called hemoglobin. Hemoglobin is found in the red blood cells and is responsible for carrying oxygen through the bloodstream. The hemoglobin tetramer consists of 2 alpha subunits and 2 beta subunits. HBB is encoded by the HBB gene on human chromosome 11. Sickle cell anemia is a recessive genetic disease, which means that both parents must be carriers and pass down the HBB gene for this disease to be expressed in the child. The gene on chromosome 11 codes for the mutated beta globin when creating the red blood cells, therefore causing sickle cell anemia.
Due to modern medicine, sickle cell anemia is no longer an effective way to fight against malaria. In fact, it causes a higher risk of death from blood clotting and other health problems. The people who are still affected by sickle cell anemia now get treated using modern medicine. Some ways to treat this disease is regular blood transfusions, therapy, and daily antibiotics, and if severe, the patient will be prescribed hydroxycarbamide. This stimulates the body to produce another type of hemoglobin called foetile hemoglobin. This hemoglobin is not affected by the mutation and substitutes for adult hemoglobin to carry oxygen around the body.
Finally, modern medicine and scientists have solved the issue around this disease caused by a random mutation. There are methods of treatment, but no cure.